So in this diagram we can see we have our DNA, and in this little yellow box we have what's known as the cleavage and polyase signals, which are encoded in the DNA. And what this will do is when this little section is transcribed, it will signal the release of what's known as CSTF, or cleavage stimulating factor, and CPSF, cleavage and polyadenylation specificity factor, from the RNA polymerase tail onto the nascent mRNA. And then so down here we can see our nascent mRNA, and attached to the RNA polymerase tail we have our CSTF and CPSF. And so what happens is, when that little cleavage polyadenylation signal is transcribed, that signals the CPSF and CSTF recognize that, and then the nascent mRNA is cut off, and then it moves over here. And so now here at the now free nascent mRNA, a protein known as poly A polymerase, or PAP, will come and bind to and add alanines to the 3' end of our nascent mRNA, and it'll add about 200 alanines at a time. And once, you can see here in pink, the alanines are added, another protein known as poly A binding protein comes and binds to the newly added alanines, and it's not really sure what the point of these proteins is, but it's believed that they help determine the length of the poly A tail. Maybe.